Today we're speaking with Dr. Charles Perou, Professor of Genetics and Pathology and Laboratory Medicine and member of the Lineberger Comprehensive Cancer Center at the University of North Carolina at Chapel Hill. His talk was Next Generation Sequencing for Gene Expression Profiling. Thank you for joining us.
My pleasure.
Your lab uses genomics and genetics approaches to characterize the diversity of breast cancers. How do you utilize the next generation sequencing to identify disease subtypes and their unique biologic features?
Well, over the years, we've been doing DNA microarray profiling of breast cancers and identified disease subtypes with that technology. And now we're switching to this newer technology, which is even more powerful, more quantitative, and provides more information. And so we're in the process of doing larger sample sizes, and our expectation is that we'll see the same disease subtypes we saw before, and possibly new subtypes, because we're collecting information types we didn't have before. And certainly we'll expect to see further refinement of the existing subtypes and possibly identify new and rare subtypes, again, that we didn't see before.
Would you speak to what you've seen with the basallike subtype?
So in the basallike subtype, as many can appreciate, this is a quite heterogeneous disease, and we're seeing many different changes within that group that are unique to that group. And a couple of the examples I showed in my talk were, and actually another investigator, Andy Futrell, also showed in our talk, is that this subtype has a very high frequency of P53 mutation. And so one of the quite exciting features of the next generation sequencing technology is we can see that on multiple levels. So we can see that when we sequence the genome of these patients, as was done by, again, Andy Futrell and his group at the Sanger, and Elaine Martis and her group at Washington University. And then our group also did this using from the gene expression profiling data, we could see the mutations in that gene. We also saw some interesting, what were called fusion proteins, where in this particular instance, a large genomic deletion occurred in the basallike cancer. It brought together two genes that were normally separated and made a hybrid between the two. And one of those two genes was an important regulator of the cell cycle, and now that regulator is clearly dysfunctional, and that may be contributing to that basallike breast cancer.
You said that the more we study breast cancer, the more we discover how complex it is. Does next generation sequencing help sort through large sample sets in order to enable you to ask the important questions about the smaller subsets?
Well, it will certainly help us figure out the complexity of breast cancer, because now we're getting an ever finer look at breast cancers. The analogy is kind of like using a black and white TV before, and now we have an HD TV, right? So we can see more of the picture. The challenge, though, is that as we see more of the picture, we may even need larger sample sizes to really be able to effectively utilize this additional information, and to find all the key information always requires large sample sizes and well-defined clinical cohorts with good treatment information.
How does this translate to the clinic? Can gene expression associated with different subtypes help predict response to therapies, and is it useful as a diagnostic tool?
So we certainly think that knowing your expression subtype or your intrinsic subtype it provides important biological information, important prognostic information, and possibly predictive information. And there are other, obviously, gene expression tests available for breast cancer patients. So breast cancer or gene expression is providing valuable information in the clinic. Now, the next generation sequencing data is even going to provide additional information that we're not getting with the gene expression. For example, the presence or absence of mutations, right, which we can see this in the gene expression version of the next generation sequencing data. We can see it in the DNA sequencing data. The presence or absence of these fusion proteins. The other real powerful advantage of the next generation sequencing is the ability to identify and quantitate alternative splice forms of different genes. This will probably also provide important prognostic and predictive information. And so this is some of the real attraction and excitement about the new technology.
Dr. Perou, thank you so much.
My pleasure.